Okay, so we're going to call it the seen and unseen benefits of bats to humans. Next slide. These are bats that are found in the U.S., not necessarily all in this area. There's only one bat that's not found in this area, and this is this bat. This is the pallid bat. It's found in desert regions of the Southwest. But we have a red bat, and we have a silver-haired bat, a big brown bat, little brown bat, hoary bat. No freetail. And that's why I was going to get rid of that pallid and put in a freetail. So 16 species of bats in Tennessee. They all eat insects. They all find their insects by echolocation, right? They have different echolocation calls. The calls differ in frequency and temporal characteristics. If I turn this switch, does the light come on? Aha! I did a crude diagram here of the echolocation calls. Can you see that? Of the yeah, I've got to make it easy on you guys that are trying to film this. Of the echolocation calls, frequency is on this graph, time is on this graph. And this is frequency. Frequency is in thousands of hertz, kilohertz. Time is in milliseconds. So very high frequency, very short duration calls. We have here a bat that starts to echolocate in the high 20s. The frequency modulates, the frequencies go down and down to about 18 kilohertz. Okay, that's a free-tailed bat or a hoary bat. Hoary bats have very similar frequencies. We have one or some here that start in the mid 40s and swoop down into around 30 or so. Free-tailed bat call is very long, that is over 10 milliseconds. These calls are about 6 milliseconds. And then we have little brown bats that are way up here. They'd be 60 kilohertz, something like that. Swooping down, but shorter duration, down to about 50 kilohertz. When I finish talking, which will hopefully be just around the time that it starts to get dark, we're going to be going out and listening for bats using ultrasonic detectors. You guys know what the highest frequency that humans can typically hear? Okay, the very best human hearing will cut off at about 20 kilohertz. And those would be children typically. There are some oddball adults that retain that high frequency hearing, but most of us are hearing down in this range, 500 hertz, not even 10 kilohertz, 5 kilohertz, I'm sorry, 5 kilohertz. As you get older, your high frequencies drop out. So bats start to echolocate at about where our hearing drops out, where we lose our hearing. But they use the same apparatus. Thank you. Everybody see the dead bats? They use the same apparatus that we do, same apparatus that I'm using to talk and the same apparatus that you're using to hear. So they're mammals, of course, you know that. And yeah.
with these guys, so the different sounds, so the little brown is that the smallest size as well?
No,
Okay.
no.
I was wondering the frequency versus size for like vocal boxes and.
Really great question. Great question. There is a rough correlation of size of bat and frequency that they use. So smaller bats tend to use higher frequencies, shorter wavelengths. Larger bats tend to use lower frequencies. But it's not really the size of the bat, but the size of the prey that they eat.
Yeah.
Okay, so freetails, if you looked at, we could go back and show that beautiful picture again. They have a huge mouth. And this is part of my story. Their main diet tends to be moss and large moss, good-sized moss. So they're a relatively small bat. They've got a wingspan of about like this. They weigh 12 grams, half an ounce. Okay, but they eat big things. They have a lower frequency. Hoary bats do the same thing. They eat big things. They're a bigger bat, much bigger bat. So, yeah.
Yeah. Cool.
So the other thing this will come up again in just a few minutes, but let's say we have a bat here. These are what we call search calls. The animals are out there at night looking for something that's flying, something in the atmosphere that they want to eat. And so they send out these search calls, which tend to be the longest, lowest frequency calls that they give, because the lower the frequency, the longer the call will transmit. So the lower the sound frequency, the longer the farther it goes before it's attenuated or loses energy in air. So this bat would be going like, the change in frequency as it goes on. When it detects something it's interested in, it shortens the call and increases the frequency modulation. So it goes, and when you hear bloop, it's the death of an insect. Okay, so, and what they do by increasing the frequency modulation, they're decreasing the distance that the call travels, but they're getting more information. Okay, getting more information to say this is something that I wish to eat. Okay. So these are the insect-eating bats of Tennessee. How many bats in the U.S., the continental 48? Come on.
120.
120. Down, less.
58.
Close, 46. 46. Okay. Next slide. How many bats in the world?
122.
200.
How many? Come on, speak up.
200.
More.
800.
More.
1,200.
More. So I got to tell you, when I started doing bat research, and my first research on bats was in 1977. There were 960 identified species of bats. There are now over 1,500 worldwide. Now, these are not rapid speciation events. This is because bat fauna is not that well known. Okay, so this is a just an example of some of the worldwide diversity of bats, and it is also illustrating some. I mean, they're just a great fauna, you know. I mean, the smallest bat. I don't think it's there. The smallest bat is the bumblebee bat, Thailand.
The one in the middle, in between, on the far left side.
Oh, this one?
Yeah.
No, that's a Glossophaga. That's a small bat, though, but it's not the smallest of them. This is a pollinating bat. Pollinating bat. Carnivorous bat. Fish-eating bat. And a bunch of fruit-eating bats.
How do they do echolocation with fish?
Pardon?
How do they do echolocation with fish?
How do they do echolocation with fish? They fly low over the water and they echolocate and they detect the small ripples that fish are making when they're near the surface. They then drag their huge feet through the water and gaff them.
Wow.
This is a neotropical bats, Mexico, the Americas, Central America, all through the Americas, exclusively neotropical. Great bat. Yeah.
I'm just wondering, like, yeah, a fruit bat could use echolocation to find a fruit, and an insect bat can use echolocation to find an insect, but they probably have preferences for what insects and what types of fruit they hunt for. So how did they discern differences using echolocation over shapes that are similar sizes?
So, first thing is that bats like this spear-nosed bat or that spear-nosed bat, those are neotropical fruit bats. They are what we call whispering bats. Very, very weak echolocation calls. They're not looking for insects. They're looking for fruits or flowers. And so they echolocate. They're not aerial chasing insects. The insect aerial chasers, and I didn't mention this, but do you know what the sound intensity is of coming out of one of these bats? I mean, it's incredible if you could hear it. If you take a free tail, which is the one I know best, if you take a free tail and you are, I think it's 100 centimeters or 10 centimeters from the bat's mouth, it's 120 decibels, which is like a smoke alarm. So we can be really happy we can't hear it. So very, very intense calls. The fruit-eating bats have very much softer calls. They do echolocate, but now this bat doesn't echolocate at all because it is a flying fox. It has the great name of Hypsignathus monstrosus, and it's an African bat that's a flying fox. Here's another flying fox, typically known as Old World fruit bats. They have large eyes. They find their way around mostly with vision and smell, and they don't echolocate. Some of them echolocate by going like that, but they don't have the very sophisticated echolocation abilities that. But to elaborate a little bit on David's question, this bat, which has a long tail of almost constant frequency, okay? What this bat is sensing in this constant frequency is slight glints from the Doppler shifts as the insect it's chasing his wings are going back and forth, back and forth. So the bat is in essence able to almost, to some level, key out the insect that it's chasing by wing beat frequencies. We don't know all the information. This, I mean, it's phenomenal, but we don't know all the information that the bats are getting. I certainly don't. Okay, next slide. So there are pollinating bats. About 75% of those 1,500 species, 75% of those 1,500 species eat insects. Okay, most of them eat insects, primarily insects. Some eat a combination of fruits, insects. Some are truly omnivorous. There are pollinating bats. They are, of course, adapted to pollinate flowers that bloom at night. And a lot of cactuses bloom at night. So this is a Mexican long-nosed bat. And it's bats of the desert Southwest with a long nose and a very long tongue to swoop down into the flower. Next slide. Yeah, so cactuses are very common, but when you go into tropical areas of the world, there are bunches of other plants that are Chiropterophilus that friendly to bats that bloom at night. Kapok trees, silk cotton, they're all flowering at night. Next slide. Tequila. Tequila is made from blue agave. Blue agave plants are pollinated from bats. Mm-hmm. Yeah. Most tequila these days is made from cloned cactuses, but there are only a couple of clones. So when you buy a tequila, you should be buying bat-friendly tequila, which are the growers that are allowing a certain percentage of their plants to flower and be cross-pollinated so that there's more genetic diversity going into the agaves, so that we don't have, like the Irish potato famine was caused by all of the potatoes being a single genotype. The bats are diversifying. Okay, next slide. There's over 500 plant species pollinated by bats. Kapok trees, bananas, mangoes, guava, durian, cocoa. Okay, all at least originally natively pollinated by bats. Next slide. Yes. Bats with pollen on their faces, so they get it on they have long tongues that lap up the nectar, and then the pollen, of course, attracts on their face, and they carry the pollen from plant to plant. Next slide. Seed dispersal. A lot of bats, particularly in tropical regions, I was going to say particularly, 1,500 species, 46 species in the United States. How many species do you think are in Canada?
46.
12.
11. How many are in Mexico?
More than 46.
Mexico, get the trend?
300.
How about 137? Probably more. Yeah, how many in Venezuela? 200 and something, okay? And then you get down to Chile. How many in Chile? 10. 10. So, bats are tropical. Most bats are tropical species, and so people that live where we live don't really tend to know bats as well as people who live in tropical areas. The highest species diversity in the world in bats is in the neotropics, so Venezuela, Colombia, these are the super high diversity areas in the world. And this is followed by tropical Africa and Asia. Seed dispersal is very important. The bats pick up the fruit and carry the fruit and drop it, but they also, next slide, they also eat the fruit and then poop out the seeds. In tropical regions, this is piper, which is a pepper plant, and they're like little, they stick up, attracting bats. They're out there to attract bats, okay, designed to attract bats. So, advantages to bats, things that bats are doing: pollination, seed dispersal, and of course, next I think, insect eating. And here's a free tail with the corn earworm moth. And this was work that we did for a number of years in Texas, in agricultural regions in Texas, where free-tailed bats are very abundant and where they grow a lot of corn and cotton and row crops. That's the corn earworm, and that's what it's called when it's eating corn. Cotton bollworm is what it's called, the same insect, when it's eating cotton. Tomato fruit worm when it's eating tomatoes. It's a polyphagous insect, and it is the #1 agricultural pest in America. Over $1 billion annually in lost production and use of pesticides to control them. Okay, and free tails, a lot of our work has been looking at the value of insectivity. Next slide. So here's the bat, and here's the moth. The moth is being released. This is again an experimental. There we go. There's the moth. There's the bat. Missed it. And there's the diagram of what's happening. So this is what you guys will be hearing when hopefully, no guarantees. No guarantees. The animals will do what they want, but this is what you'll be hearing when we go. We'll do it once more because I can see who you're rooting for. So you hear the rapid succession of pulses as the animal's approaching and eating the insect. Next slide. Okay, so where are we at time-wise?
7:45 on the dot.
What time should we be out there? 8:15? 8:30?
Sunset is 8:43.
So dusk around 8:15.
So we got plenty of time right now, still, yeah. Okay, so I'm going to talk a little bit about some of our research, stuff that by I mean me, my colleagues, and my PhD students, of which there were some 30-some students, doctoral and master's students who worked on this project. Okay, hypothesis. Bats, next slide. Bats go to insects. These are bats leaving a roost in Texas, snaking out in large numbers. This is a helicite, which is a hybrid kite balloon, helium balloon with a kite attached to it, which we use for sending up equipment. And that equipment can include a insect net, which is obvious here, and bat detector, which is not so obvious, as well as other gear, which tells us what the altitude is, what the wind speeds are, all kinds of cool stuff like that. Bats go to insects, and why? Okay, next slide. What do we know about insects aloft? So I'm going to switch to the insects perspective. What do we know about insects aloft? Well, fairly recent work on insect migrations. And a lot of this insect migration research has depended on radar. It's been known for a long time that insects migrate, but it's not been known where and how they migrate. Okay, so this is a paper published in Science in 2016 by colleagues that we worked with. Next slide. Now this is over Great Britain, southern United Kingdom, but it's been estimated, and this is from radar work, that at altitudes of 150 to 1,200 meters above the ground, above ground level, that there are about 3,200 tons of biomass, insect biomass, that migrate annually, 3.5 trillion insects. For perspective, this is 8 times the total biomass of the migratory songbirds that migrate annually between the UK and Africa. Okay, huge insect migration. Next slide. Believe it or not, I do. A greater diversity in biomass migrates in the lower atmosphere of the Earth. We're talking 100 meters above, 100 meters to 3,000 meters above the Earth, than walks on the surface of the Earth. Okay, it's phenomenal, the biomass. A lot of this biomass, a major proportion of it is insects, but it also includes birds and it includes bats, other flying animals. Next slide. So, and this is coming from that slide, it's just showing that the insects go aloft to catch prevailing winds. And these prevailing winds tend to be seasonal. So if you're in a temperate area, say North Temperate Zone, in the springtime, those winds develop and they go from south to north. In the fall, they reverse themselves and go back. In the fall, it's easier. They come back on cold fronts, the developing. Okay, so this is just saying that the insects migrate both in the daytime and at night. So it's food for both birds and bats. And this is the sort of instrumentation that's used. This is altitude, and this is percents of total of medium and large insects during the daytime, dusk, and at night. Okay. What we were looking at night, large insects at altitudes of 400 to 700 and 800 meters above the ground. Next slide. This is the Doppler. I got into this project in 1995, the same year that they turned on the Doppler radar in New Braunfels, Texas. And the Doppler radar in New Braunfels is 18 kilometers from Bracken Cave, a colony of several million free-tailed bats. Okay, and I called this guy Jim Ward. I forgot his name. The guy that was the manager of the New Braunfels Doppler radar, and I told him that I thought this would be a wonderful tool for studying the behavior of bats at aloft. We know that the bats go up high, and we didn't know how high or where, but this would be a great tool. Jim Ward said to me, well, if you want to study bats aloft, you should connect with John Westbrook and Wayne Wolf, USDA researchers, because they are studying, they want to use the Doppler radar to study insect movements. So Jim put it together, and so did we. So this is a plot from my friend John Westbrook. This is the Doppler radar in New Braunfels, not New Braunfels, this is in Brownsville, Texas, extreme southern tip of Texas. And so this is the Rio Grande River here. The radar is located here. This is 9 o'clock on a clear night. This is 10 p.m., 9 p.m., 10 p.m., 11 p.m., and these are moths. These are moths emerging from agricultural regions in the lower Rio Grande Valley of Mexico in the U.S. And these, and my friends, my now good friends, were working for the USDA, and their job was to predict when major agricultural pests are going to arrive in agricultural fields so that they can kill them. Yeah, so they can protect the crops, right? And this is the sort of images that they saw. And this is the vector movement of these moths. So estimated 9 billion corn earworms and 7 billion fall armyworms, which is another nasty pest of crops. Okay, that's the insect side of it. Next slide. There's free-tailed bats emerging from Frio Cave. Was estimated at one time to be 12 million bats in Frio Cave. Nowhere near that many now, but still many millions. Next slide. This is the Doppler radar in New Braunfels, Texas. So how do Doppler radars work? They work by detecting movement, movement in the atmosphere. To a Doppler radar, a free-tailed bat has the reflectivity of a small hailstone, and insects have the reflectivity of light rain or mist. This radar is again about a little over 30 miles from Bracken Cave. Next slide. So here's where we were working. This is the agricultural region. Okay, there's the Brownsville radar. Here's the New Braunfels radar. This is Bracken Cave, estimated 20 million bats. Ney Cave, 10 to 12. Frio, 12 million. 4 to 6 in James River, 4 million in that Congress Avenue Bridge. Okay, those are your reference points. Check these reference points and do the next slide. Next slide. The halos are bats emerging from each of those roost sites. To the Doppler radar, the bats look like a tornado erupting from ground level and spreading out over the landscape.
That was fun the first time.
Oh, God, was that fun.
Holy cow.
Oh, God, was that fun.
So they weren't picking up bats aloft when they were studying insects before you approached them?
Question, I don't understand.
Yeah.
Say it again.
So initially when you described how you met these other scientists that work for the USDA, they were tracking insects, and you were interested in tracking bats. They weren't picking up on the bat Doppler activity.
You could not help but see the bat's Doppler activity, and that's how I got tripped into it. Is that I was told that this is what they were seeing on clear nights, and we knew what it was.
Right. Okay.
Now, but we were not the first people to actually look at high-flying bats. All you have to do is sit out in front of one of these caves and watch the bats go aloft, and then watch them with your binoculars. When they go out of sight for your naked eye, look at them in your binoculars, and they go out of sight in your binoculars. We knew they were going aloft, and in the 1960s, there are 3 air bases in the vicinity of San Antonio. And in the 1960s, there were T-38 jet trainers.
Yeah.
20% of the nighttime flights of T-38 jet trainers came back with some damage. Several of them lost engines because even something the size of a bat it's not like you run into a golden eagle or a vulture or or a goose. But so it'll splatter on the windshield, but it can startle the pilot. But if they get sucked into the afterburner, they kill the engine. So the Air Force knew that there was a problem. They commissioned a radar study which was published in the 70s, which showed that the bats went aloft. But they didn't know why the bats were going aloft. We didn't know why no one knew why the bats were going aloft until we put it together with the USDA researchers. Yeah. Next slide. So, lots of papers now linking migratory insects and bats. Anyone wants reprints, let me know. I'll send you some PDFs. Next slide. And I've spent a good part of my career studying bats and insects aloft. I already introduced to you the helicite. This is Jennifer Crowell and myself launching one. Our bat detectors were put inside of these cylinders. We had insect nets, and we used radar, both types of radar, and we developed fecal DNA probes so that we could look at the poop of the bats and tell you what the bats were eating. Okay, so fecal DNA was a marvelous tool that only became available in the mid-90s. Next slide. DNA of the insect survives in the bat's poop. There's bat poop. Next slide. This is the availability of corn earworms assessed by our agricultural friends using pheromone traps. So highly seasonal peaks. This is the migration of the animal, of the moths in spring. They infest corn. 18 days later, they leave the corn, they infest cotton, and then they continue on their way north. Okay? The blue is the abundance of corn earworms in the bat's poop. So it's a correlation, but a strong one. And it's not only a correlation because we had the fecal DNA evidence. Okay, next slide. There's new techniques now for looking at DNA in environmental samples. Metabarcoding allows us to sample not just for corn earworms, but for all of the insects that are out there. A recent student of mine, Jennifer, found the DNA of 44 species of pests in the diet of the bats, free-tailed bats, and at least 21 species of migratory insects. So, major pests. Next slide. This is a bat immediately before and a couple of days after the insect migrations. So, before, after.
Chunker.
And these migrations could not be timed at a better time for the bats, and it's probably no accident. The spring migration occurs just as these huge colonies are maternity colonies. Females are aggregating to have their babies. Yeah.
So, did you shave bat butts?
Did we freeze bat butts?
No, did you shave them? Like,
Shave them?
Remove the hair.
No, we didn't shave it.
They look very.
No.
It kind of looks like,
No.
you're like, oh, let's shave it before.
They took him to the barber.
But good question. We're not above it.
I know, that's what I was
And then I think we can go back one slide. No, 2 slides. I didn't mention it at the time, but this is September, October, when there's another spike in insects, migratory insects in the bat's poop. And this was really fun. The USDA had seen it too, but we only looked at this because this is what's agronomically significant. By this time of the year, it's hot and dry, and not only are the crops shriveled up, but all the local vegetation, the native vegetation, is really shriveled up as well. And the bats are basically surviving on occasional thunderstorms coming through and alate ant emergences and stuff. This is the arrival of the first cold fronts. So this is when the bats are giving birth to and raising their young, and this is the time when the bats are getting ready for migration. Okay. Go for it. Evaluating. So the next stage is, what is it worth? What's all this insectivory worth? So we have a number of papers in which we evaluated the economic value of the pest control services of the bats. No one else had ever done this one, but next slide.
Hold on. I got to take a picture of that because I need to ask you for some of those.
Okay.
That's,
So I can always send you a PDF too.
yeah.
These are the empirical papers that we wrote with the modeling group at UT. Tom Hallam, Paula Federico, these are brilliant modelers. This is mathematical group, and they were, I mean, that's how science is done, right? Collaboration. I mean, and these guys were just great. So these are their empirical papers of what's the value of the bats to the cotton farmers in Texas in terms of crops not eaten by pests and pesticides that they don't have to spray on the landscape. Yeah.
I teach a whole unit on agriculture, so.
Huh?
I teach a whole unit on like soil and agriculture,
Uh-huh.
so that's why I'm like, and
Oh, okay, cool.
Need some of
Yeah,
that. Let's go.
it's all right. It's
Yeah.
okay, next slide. Whoops, and then I had this absolutely brilliant postdoc, Justin Boyles, and Justin elaborated the value of the bats in our small area to every county in the U.S. for which he could get information. And his estimate of the annual value of bats to U.S. agriculture is somewhere between $3.7 and $53 billion annually. And somehow we got this published in Science. So,
sweet.
okay, where am I at? Next slide. Okay, that's basically one species of bat that we looked at and one crop type. Now, Justin's elaborated it, but just think about all of the 1,500 species of bats scattered across the globe and what those contributions must be worldwide. Next slide. And there are people who have been doing it all over the world. Establishing the value of bats in other crops in tropical regions, not only in corn, but rice is a big one, neotropical agroforestry systems. And next slide.
What is the,
Yeah, I'm sorry.
what is the relationship with the bats and the corn?
The corn earworms
Oh, okay.
are the major pests to corn production.
Okay.
Yeah. And now, it's the larvae of the insect that eats the corn or that eats the cotton. Mm-hmm. The bats are eating the adult insect and hopefully, ideally, eating these insects before they lay their eggs. But it doesn't matter. It does matter because each insect lays 1,000 eggs. Yeah.
It still matters. It still matters.
Okay, so next slide. I think that's just more, yeah. And documenting contributions to food security, ecosystem integrity throughout the world. And that may be it. Let's see. Yeah, that's it.
Wait, hey Garrett,
Yeah.
what about that paper you're telling me you recently published about like feces over Texas?
Ah!
What's that one?
So I've been retired for like 4 years, but I had a backlog of papers that need to be published. I still do have a small backlog, and the one that I really wanted to get out is titled "Fecal Rain Over Texas."
Nice. That's a good name.
Yeah. And I knew the Journal of Mammalogy. I knew one of these hoity journals would make me change the title. So I sent it to a journal called Acta Kuraptologica. It's published in Poland, but I know the editor, and I knew he had a sense of humor. So it's been said, people are looking at the ecosystem services of bats, what bats do for humans, and it's been hypothesized that bats perform a service by pooping nutrients. You guys were talking about nitrogen and phosphorus in the water. What's the nitrogen content of bat poop?
60%.
It's not that high, but it's high. Yeah, it's 12 to 14%, but it's pretty high though. It's really strong fertilizer,
Yeah, nitrogen.
and it's got high phosphorus and also potassium. So it's great fertilizer, and that's been known for years. They've been mining guano. They mined 100,000 tons of guano from Carlsbad Caverns between 1901 and 1926. 100,000 tons of guano. Anyway, fecal rain over Texas, or nutrient distribution by bats over an agricultural landscape, which is the serious title. But I got fecal rain over Texas in, and I was going to make Greg Abbott a co-author, governor of Texas. But I didn't know if I'd get that one through. But anyway, the answer is, and there have been other people who have hypothesized this, but no one had the right system. There had been one other study that had done it, but they'd only caught five fecal pellets. They said you need bigger traps, more bats, bigger traps, more bats. Well, we had it. We had the highest bat foraging densities reported anywhere in the literature. We built gigantic fecal traps, and we put up a lot of them. And I got to tell you, they don't contribute very much. There's more nitrogen deposited as atmospheric nitrogen than there is from bat poop. So is that story. But you maybe talk about it. Yeah. Any questions? We had lots of questions about.
Maybe just briefly, the premise of the project is that the statistical tests that are common in science can be sometimes hard for kids to learn. So the null hypothesis statistical testing. My question is, can you talk about the role of that kind of statistics, for better or for worse, and more generally how you think about how you build the case for some argument? Do you use statistics? Do you use evidence? How do you?
Oh gosh, yes. Yeah, I mean, let's talk about fecal rain over Texas. My working hypothesis, and the working hypothesis of lots of researchers, was that this was going to be a significant service, particularly in areas where there's a lot of bat poop coming down, where there's a lot of bats. So I set up, I guess, the main year. So we did this over a series of years as a lab project. I've got all these kids out there working in Texas, and so we start figuring out what's the best fecal trap, and we finally get the right fecal trap, and we're catching poop, and we can then extrapolate how much is dropped every night over a hectare. And then we take that, and we know, so we know how many feces, and I can't repeat how many feces are dropped on average per hectare. And it differs, depends on how far you are from the cave and if you're in an agricultural region or not. But we take the best case scenario, and then we do a statistical test. And we do a statistical test, and we find that it's not significantly different than zero. I mean, and we compare it against the atmospheric distribution of nitrogen. We compare it to what the recommended applications are for bat poop as fertilizer by commercial bat poop salespeople, and compare our results. And yeah, is that getting at?
Maybe it's really briefly, because I'm sure others have questions. Like, what about not comparing the amount you got to a certain quantity, like 0, but just trying to figure out how much poop is there? How do you get a good answer to that question about, like, this is our estimate across Texas?
Well, the question of how much poop is there is not so straightforward. We put 4 of our fecal traps right outside the cave, within 50 meters of the cave, and then we took a corridor, a riverine habitat corridor, and we spaced our traps out. And then we got into agricultural region. So we had distance effects, we had habitat, and we had, well, distance from the cave, natural habitat features, and we had agricultural. And we also had, we have a system down there now where genetically modified crops have come in a big way. So we're also interested in what are the impacts of genetically modified crops on bat activity, because they're supposed to kill all the insects. Mm-hmm. So we have all of those variables that we look at and see. And there's a strong pattern of distance. There was no habitat association at all, much to against our expectations. But distance we expected.
Thank you.
Yeah.
Yeah, really interesting.
Nice. Good question.
Oh, Audrey had a question I know that she was keen to ask, but she's stepped out.
I think she had to go home with her daughter,
That's
but she's coming back.
okay.
Anybody have any questions about bats?
I have a question about the free-tailed bats.
Sure.
So they're not native here, right?
Oh, they are now.
They are now, but do like when we have other species that come in, they tend to compete with the local. Is there much competition or?
You know, presumably, but, and the paper was just published, pins it totally on climate change, of the expansion of the bats, totally on climate change. But we've also had the confounding variable of large numbers of bats having been removed over a similar time frame by white nose. So we have another species of bat, which the big brown bat, which is mildly affected, but not seriously affected by white nose. And big brown bats are also become much more abundant. So is it competition with, is it out competing with other bats? I suspect it is, but we've got so many variables. Yeah, good question. Yeah.
I have a question.
Sure.
In the course of your work, have you uncovered a new species that maybe was not documented before in science?
No, I haven't. And I have not, I have sunk a species because there used to be thought, it was thought that there were 2 species of freetails. One of them was Taderida brasiliensis and the other was Taderida cynocephala. It was thought that the free-tailed bat that lives in the eastern, that was living in Florida, Georgia, Louisiana, was different than the species that occurs in the south central U.S. and down into Mexico. But they're not. They're the same species, same gene pool, which translates to being the same species. So we sunk one. Yeah.
You came in and told my class in the evolution session about like how you uncovered the difference of the evolutionary pressure for parents to take care of their young.
Oh,
Would you want to speak to that briefly before we go outside?
well, we're going to have to go outside soon, but
Yeah.
So, not a subject of this talk, but so I mentioned that these bats aggregate in these colonies to give birth to and nurse their babies. So, the females, they mate in Mexico or on the migration routes. They come up to these large cave roosts and they give birth. Each female gives birth to a single baby. There's occasional twinning, but no more common than it is in humans. And they all give birth at about the same time, within a couple of week period. And they then, and they're mammals, of course, so they nurse their young with milk, as do all mammals. So it was published in the 1960s and repeated as a textbook example that when they after they give birth to their babies, they put the babies all together. And they put the babies in certain parts of the cave. These tend to be high parts where the caves are very hot. So these are, and the babies are naked. They're always naked, but they're furless. They're furless, and they cluster together to stay warm. And they put the babies in these dense clusters to where you have an area, a meter square, you have 5,000 babies. And each baby has a different mother, okay? And it was stated in the literature that when after the mothers gave birth, they clean their baby, they give it a milk meal, and they put it in what they call the creche, these dense, that the mothers could not or did not find their own baby because that's what it looked like. The babies are very aggressive. The babies just want to be fed. Females land on the creche, they get attached to baby, they have to fight off babies. So it was published that this was random nursing. And that was my first NSF grant. And that was just after I took my job here at Tennessee. I had, before that, worked on bats and had done genetic studies looking at paternity and maternity on bats in Trinidad in the West Indies. I saw this example of apparently apparently random nursing, which makes no sense. Anybody want to tell me why it makes no sense?
Burn a lot of energy making breast milk. You want it to go to your baby.
You use a lot of energy making breast milk. So if you take a typical mammal, and humans are typical in this sense, and you look at the amount of energy that a mother puts into a child from conception to birth versus birth to weaning, presuming that they breastfeed. Do you know what the breakdown is?
Twice as much?
5 times as much.
Okay.
Yeah, 5 times as much energy is put into young from birth to weaning. And if nursing was truly random, and this was my argument, in the NSF grant that I got funded for. If nursing was truly random, what would the smart mother do?
Never show back up.
Pardon?
Never show back up.
Never show back up. You could have got the grant. Yeah. Or if you want to put it in the jargon of the day, it was an evolutionarily unstable strategy because it was invasible. It could be invaded by the mother that never showed up. And the argument at the time when they published this was there were common, and you still hear these arguments, that something's good for the group or good for the species. Evolved because it was good for the group. And they said, this ensures that the most aggressive, strongest babies get fed. But from a mother's point of view, it's an evolutionarily unstable strategy. And that's what I argued. And the reality is so what we did, again, being simple-minded, was just to go into the cave when mothers are nursing babies and pluck a mother and the baby she's nursing, take a small blood sample from both of them, and then put them back. We didn't know who she was nursing before or who she would nurse, who she nursed yesterday or who she's nursing tomorrow. We only know that she's nursing that baby now. And then we did their genotypes. Back then we had to do their genotypes with some more primitive tools with protein electrophoresis. But it was clear that mothers' genotypes were associated non-randomly, which meant that, nursing was non-random. Didn't necessarily mean that mothers are nursing only their own babies. They might be nursing their own baby and somebody else's baby to whom they are related. That's still out there as a possibility. But we also found in almost 20% of the cases, mothers were nursing a baby who couldn't be their own. So they are losing milk. But the difference between milk stealing, milk thefts, and mother-baby interactions are dramatically different. When a mother, and we watched this with infrared video cameras, and when a mother is attacked by a baby, and the baby latches out to attach to her teeth, she flaps at it and knocks it off. When she finds her own baby, she nuzzles it and brings it in, and they nurse for a good, 10 minutes or so. Yeah. Yep. That's my good mother talk, which I tend to give to garden clubs of elderly ladies.